Hi, Michelle Glass here. Welcome back to another one of our Chapter 16 video lectures. We are working in Chapter 16 on the autonomic nervous system, which is, of course, the unconscious regulation of your viscera, your visceral organs, right? So that's your smooth muscle, cardiac muscle, adipose tissue, and your glands. We've seen the two primary divisions of the autonomic division are the sympathetic and parasympathetic. We have a whole video already addressing the sympathetic division, which is our fight or flight. And this video is obviously focusing on its companion, which is the parasympathetic. And we can think of it as basically being the opposite. So here our catchphrase is rest and digest. Okay, so just like in the sympathetic division, we need to pay attention to the same type of information for parasympathetic. We need to know how it's organized. We need to know the basic functions. We also need to be familiar with both the neurotransmitters that are released and then the types of receptors that those neurotransmitters will bind with. So let's look at organization first. So here we have a nice summary of the organization of the parasympathetic division. Now, right away, you should notice something that's opposite. In the case of the parasympathetic division, our preganglionic fibers are super duper long. These preganglionic fibers are long because they are going to be synapsing onto ganglia, which are going to be located very close to the target tissue or to the effector tissue, or actually embedded in the wall of those organs. So these preganglionic fibers are extending from either the brain stem or the sacral region of the spinal cord, going all the way out to those effector tissues of the body. Now, when we look at the preganglionic fibers that are coming off of the brain stem, we see that they are going to be making up 4 of our cranial nerves that we studied last semester. So we will see the oculomotor nerve 3, facial nerve 7, glossopharyngeal nerve 9, and vagus nerve 10 are all going to be part of the parasympathetic division. Now notice that I have highlighted the vagus nerve 10 because 75% of the parasympathetic outflow, meaning information leaving the brainstem, going out to the effector tissue, is traveling on the vagus. So it is the super duper important nerve in the parasympathetic division. Notice also then that those pelvic nerves, which are going to be coming off of the sacral regions, are coming off of sacral regions 2 to 4. So sacral nerves 2 to 4 are going to be making up also part of the parasympathetic division. Now we've said that these preganglionic fibers are super long because they're synapsing at the ganglionic neuron, they're synapsing at the ganglionic neuron, which is located very close to the effector. If that's the case, we call that a terminal ganglion. And then some of those preganglionic fibers are synapsing onto ganglionic neurons that are actually embedded in the wall of those organs. And we refer to those as intramural ganglia. Now, when we talk about it being embedded in the wall of the organ, that means it's actually part of the organ wall of the heart or the stomach or small or large intestine or urinary bladder. So those preganglionic fibers clearly have a far distance to travel to actually become connected to those ganglionic fibers. And then that means those ganglionic fibers can be super duper short because they're close to the effector tissue in the case of the terminal ganglia, or they're embedded in the wall of the effector organ in the case of most of the organs there in the thoracic and abdominopelvic cavity. Okay, so here is another representation of the organization. Again, we're seeing that our preganglionic fibers are coming off of the brain stem, so extending from the pons to the medulla oblongata. We see that we have the cranial nerves, oculomotor and facial, glossopharyngeal, and vagus. And remember that vagus is going to carry 75% of the outflow. Now, those preganglionic fibers that make up nerve 3, 7, and 9, you see that they are synapsing onto ganglionic neurons, which are located close to glands in the head or eye muscles. And so those would be our examples of our terminal ganglia. Now, these are listed for you in the text, but I'm not going to ask you to memorize them by name. When we look at the vagus, we see that that is going to be synapsing onto ganglionic neurons embedded in the wall of our organs and thoracic and abdominal areas. So that's going to be an example of intramural ganglia. And then the preganglionic fibers extending from S2 to S4 are going to be focusing in on our effector tissues and our pelvic organs. And again, this is going to also be an example of intramural ganglia. Now, after we understand organization, next we need to make sure we talk about the function of parasympathetic division. We've called it, in general terms, the rest and digest. So what does that mean exactly? Remember, we see parasympathetic division as a companion to the sympathetic, so it's essentially doing the opposite sorts of activity. The parasympathetic division will constrict your pupils and assist in focusing your lens. We also see that it's going to stimulate and increase digestive gland secretions. We also see it's going to stimulate those endocrine organs that are important in producing our hormones that are going to regulate the absorption of nutrients. So we'll see an increase in the production of those hormones. Not only does parasympathetic division assist in kind of slowing down metabolism and relaxing the body and assisting with digestion, but it's also important in our reproductive system as well. So here we'll see that the parasympathetic division will stimulate blood flow and the gland secretions that are associated with sexual arousal. And then this is going back to our digestive tract. We see that the parasympathetic division will stimulate the smooth muscle of the stomach and small and large intestine. So we'll increase the activity of the smooth muscle of the GI tract. It will also be involved in coordinating defecation. So again, it's going to be stimulating the tissue there in the large intestine that's an associated skeletal muscle that's going to be important in defecation. And then we will also see parasympathetic division increasing the stimulation to the smooth muscle and the urinary tract. So we're going to see an increase in contraction there. That's going to be involved in the micturition reflex. And then finally, we will see that the parasympathetic division will be important in constricting the respiratory tract, bringing that back to normal, because we will see a slowing down of our respiratory rate under the control of the parasympathetic division. In order to constrict the respiratory tract, you have to actually increase your stimulation to that smooth muscle so that it's contracting more. Let me see, contracting more so that we're actually constricting the diameter there. And then we're seeing that the parasympathetic division will decrease the heart rate, so we'll actually inhibit some of the contraction or the cardiac muscle cells, slowing down the heart rate, and then having several different methods or mechanisms to decrease the blood pressure. So this is, exactly the opposite then of sympathetic division. The parasympathetic division is releasing the neurotransmitter acetylcholine. So this is our old friend. We know it very well from AMP1. So this is abbreviated, of course, big A, big C, little h. So this is going to be the one hormone, the one neurotransmitter that is being released here. Now, that acetylcholine is going to be binding to two different types of receptors. Now, we can call both of these receptors cholinergic receptors. This term cholinergic, remember, just means that it involves the acetylcholine. So the cholinergic name is coming from the choline part of acetylcholine. Now, in these two types of receptors, the nicotinic receptor is one we are already familiar with. It is our old friend, the chemically gated sodium channel that we talked about extensively in AMP1 when we discussed skeletal muscle contraction and also when we were looking at synapses in the nervous system in AMP1. The nicotinic receptor, again, is the same thing, so we're just naming it now. So acetylcholine will bind to a chemically gated sodium channel. Sodium will rush into the cell. When sodium enters the cell is excited. So now we'll see that the cell is going to be stimulated. Now, obviously, parasympathetic division is not only exciting tissue. In some cases, we need to inhibit the tissue. And so that's where the muscarinic receptors will come into play. The muscarinic receptors use the G protein and second messenger system. So remember, this means acetylcholine will bind to a membrane receptor. This will activate G protein. The G protein then relays the signal on to maybe cyclic AMP or a calcium ion, which is acting as a second messenger. This can then either excite the cell, just like we saw in the nicotinic receptor, or it can inhibit the cell. So we see then that the cells can respond in different ways, depending on the muscarinic receptor and the G protein second messenger system at play. And that's it for now.